First of all, I would like to thank the organizers for inviting me. I'm very grateful to be here. So today I will be talking about introducing diagnostic modality into biomaterials, but I will also talk about using polymers which have inherently a diagnostic modality integrated already, and how this can be used for in vivo tracking mostly. So as already have been said, I did my PhD in Prague in Institute of Macromolecular Chemistry, and then I moved to Belgium, and now I'm actually starting my senior postdoc in the same group as I did my junior postdoc. So I have another appointment for next 3 years. So here's a little overview of materials I'm working with. I will just maybe move this to the side. So today I will be talking about polymeric nanoparticles as one part of my projects. Then I will also talk about thermogelling polymers and thermoresponsive polymers, which can create injectable depots. And at last, those are my most recent projects, I would say. I will be talking about biomaterials, which can be used for regenerative medicine and tissue engineering in general. Here is a little slide about importance of in vivo tracking, but I believe that everybody here at this session probably knows about it. So I will just say briefly what is important for me. So for example, for the nanoparticles, that's something we actually want to do in future. I will not be talking about it here, but in future we will want to also test to create a targeting group for nanoparticles, so we can maybe see how they accumulate in some specific tissue which we wanted to target. And then we could also want to see if there is some specific therapeutic response to the nanoparticles, and if we can link it to the presence or absence of the nanoparticles, which is a special class of diagnostics. We also want to test them to be used for cell labeling, and that could be also their application. Then I will be talking about the injectable depots. There we always measure basically just the dissolution rates, but we also found out, and I will be talking about this in more details, that they're actually quite useful for cell labeling as well. And at last, I will be talking about degradation rates of the biomaterials, because for tissue engineering and regenerative medicine, that's something which is very important for their application, which actually determines if the tissue engineering will be successful or not. And you can create this, if you know the degradation rates, you can create this feedback loop. You can come back to your material and adapt it so you can obtain the successful tissue regeneration. You can also see here, I write about possible defects. We always want to make sure that we have a specific 3D structure of these materials. We need to always have pores for tissue engineering. I will also talk about this in more details later. So we really want to see if the material maintain their structure, and if not, how does that affect, will this affect in future the tissue regeneration? So those are really important parts for us. So as have been said here, I'll be talking about fluorine and MRI mostly. So as you probably all know, for fluorine and MRI, the usual problem nowadays is the tracers, which are very hydrophobic. So the properties in water are not really suitable. And if you make, for example, emulsions, you don't really have control over biodistribution. So that's where the polymers comes into pictures. But with polymers, you can really determine specific interaction with the body, specific biodistribution you can design for the specific polymer. So that's very convenient. But the problem is that polymers also don't have very good properties for fluorine MRI. So that's where we came with this specific tracer. It's a polymeric tracer which is in fact water soluble. It has this difluoromethylene group here. In more details, this polymer is actually thermoresponsive. I believe that majority of the people will know what thermoresponsive means, but I will still briefly say it. So thermoresponsive polymers are water soluble at specific temperatures. And then when you cross the specific temperature, you will get a phase separation or self-assembly into nanoparticles. And this can be used in wide range of biomedical application. So the idea is that you have a thermoresponsive polymer, which have already quite wide range, can be used for quite wide range of biomedical application. And on top of that, you have the diagnostic modality in it. I will also talk about later about contrast agents for radiology, because I'm polymer chemist. I made a polymer which has this specific moiety. You can see that this is a building block for majority of contrast agents, which are now used in medicine. And we transform this, basically this moiety into a monomer, acrylamide monomer, and then from that we can have the polymers, and I will show you how that can be used for tracking of your materials in vivo. So here is a little overview of materials I used I made during my PhD. I will just very briefly go through the most important results, I would say, and then I will move to a bit more recent projects. So we made nanoparticles based on this polymeric tracer. Then we made them to disassembly upon a specific stimuli that can help you to create drug delivery systems. And at the end, we also made injectable implants, which can be used for a slow drug release. So the first very interesting discovery we obtained from these polymers is that when you make a diblocopolymers with, or blockopolymers with this fluorinated polymer, with the fluorine tracer, you obtain this some sort of very special type of nanoparticles, which we call physically cross-linked nanogels. These nanoparticles has high amount of water in their structure. The polymer inside is homogeneously spread and, let's say, physically cross-linked. And these particles also have hydrophilic corona. So you can already imagine how this is useful for us for fluorine MRI. So in theory, inside these particles, you have a lot of water. So the fluorine molecules are very nicely solvated. And that results in really beautiful sharp signals for majority of our materials. We obtain very nice signal in a magnetic resonance spectrum. And that, of course, then results in a very nice image. And also what we found out that is these polymers actually have very nice relaxation properties as well. So in another project, we created these injectable implants that was made from a statistical copolymer, so that's a bit different architecture. I will also show how blockopolymers are different from the statistical copolymers from the point of view of in vivo studies. So these statistical copolymers were made again with the fluorinated polymer. Then we also added this specific monomer, which has imidazole. We know that imidazole can be protonated below a specific pH. I believe it's around 6.5. That means that here at pH 5, all the imidazole moieties are protonated and the polymer is then fully soluble in water. So when this polymer is injected in pH 5, it gets into the body where it basically gets buffered to the physiological pH, and it gets above the global temperature of the polymer. It should precipitate into a depot and basically stay in the place for some time. So we measured in vivo based on these polymers. I also want to show you that we varied this specific monomer ratio. It's a hydrophilic monomer. So the more we add, the more water soluble the polymer became, and the higher the cloud point temperature of this polymer specifically became. So, and what it was the interesting result from this study is that we found out that this specific polymer has the cloud point temperature at 54 degrees. That means that it's way, way higher than physiological temperature. So that means that when we inject this polymer, it should be fully water soluble. It should, we kind of use this polymer as a control. And we wanted to see that this polymer, when it's injected, it will immediately get cleared away because it's basically water soluble. It doesn't make any depot. So what we actually saw, we were quite surprised that we can see this polymer even after 2 months, actually. The half-life of this polymer was 31 days in the body. So we thought that there must be something happening. So we proposed that upon the injections of these polymers, there must be some interaction with the tissue in the place of injecting. So it doesn't really matter if the polymer precipitates and create a depot, or if it's fully water soluble, it still has to interact somehow with the tissue. So to prove this, we made 4 different thermoresponsive polymers. You can see them here. And one of them, this one, actually again has the cloud point temperature above the body temperature at around 50 degrees. That means that if it's injected, it's water soluble. And we saw this polymer after we injected it, we saw it for over a month. So again, we show that this is actually the case for thermoresponsive polymers in general. And also another thing is that we found out that you can really nicely tune how long the polymer will stay in the place. It can be depending on a type of a polymer and also depending on the cloud point temperature and basically the water solubility of this polymer. So that's really nice. So to test or to check what kind of interaction these polymers does with the tissue in the place of injection, we simply measured, the first thing we did is to measure if they are actually internalized within the cells. So here on this image, you can see the green is actually the polymer, and you can see that it's actually internalized very quickly. And what we also think that it's internalized in some vesicles of the cells, because you can see it's a little bit grainy as well. So this is something we actually propose that it could be interesting to use thermoresponsive polymers in general for tissue labeling. And you could also design for how long you want the specific cells to be labeled. So this can be really nice, especially for applications in tissue engineering and other subjects. So now I will move to a bit more recent projects. I will be talking about materials which makes, again, nanoparticles. And these materials can also transform into physically cross-linked hydrogels. And then at the end of this talk, I will be talking about finally the chemically crossing materials which can be then used for tissue engineering. So let's start with the particles. Here we made 3 block polymers. So before we had the block polymers, 3 block polymers with the architecture that you have hydrophobic side chains, or let's say thermoresponsive in our case, side chains, and hydrophilic block in the middle of any hydrophilic polymer. They are known to be able to, of course, make nanoparticles because it's amphiphilic copolymers. But what they do next is that at specific concentration, high concentration, the particles can kind of create this like connection between each other. And that results in creation of these porous and physically cross-linked homogeneous hydrogels. So I will not be really talking in too much details. I made 2 groups. I'm not really sure if you see it because I see us here in the bottom. You probably see the structure, so it's fine. Okay, so I made 2 groups of polymers. The first one has a very long, you can see a very long polyethylene glycol chain and very short chains of the thermoresponsive polymer. And then in the second group, we have basically a reverse system where we have very short hydrophilic polyethylene glycol chain and very long side chains from the thermoresponsive fluorinated polymer. You can already see that these 2 groups gave us very different types of nanoparticles. So overall, these polymers are very sensitive to the ratios. They can really create, the ratio is really what matters for these type of polymers. So the first group gave us, again, the physically cross-linked nanogels, the type of particle I was talking in the beginning of this lecture. And the second type of the polymers gave us this some sort of small micelles or small nanoparticles, which can then already start associate into some worm-like structures. And then when we heated up actually this sample, we already saw that you obtain this homogeneous layer of nanoparticles. And that already gave us a hint that this material will be able to make hydrogel. So after this, we actually only focus on this group. So here you have a little overview of these materials. I will just go through it quickly. What you see here is the cloud point temperature, the temperature after where the transition happens for all three polymers, and it changes with the concentration. So at low concentrations, below the cloud point temperature, we have nanoparticles. When you cross above the cloud point temperature, the sample precipitates. And then when you increase the concentration, the precipitate can kind of connect with each other, and then they create this homogeneous hydrogels. So this is really nice. And the hydrogels only appears here above the cloud point temperature. So it's actually quite convenient that we have this drop here. So in conclusion, with, let's say, with one polymer, you can have nanoparticles at low concentrations at 37 degrees at physiological temperature and hydrogels in high concentrations. So like that, you can actually cover quite a wide range of biomedical applications with one polymer. And the synthesis of this polymer is very simple also. It's only 2 steps, so it's really, really nice. Here you can see a phantom measurements on these polymers. What I wanted to show you here, which I find very interesting, and it's actually the case often for fluorine MRI, is that in the first group, you can see that with the decreased content of fluorine, the detection limits gets worse, or it gets higher, of course, because you have less fluorine, so that kind of makes sense. But you can see that in the second group, it's actually not the case. The more fluorine you add to your polymer, the detection limit doesn't improve, and they, I would say, even get worse at one point. And this is because in the second group, we had small micelles, particles which has very hydrophobic core. So the more fluorine you add, the more hydrophobic it became. And then there you eventually end up with a loss of signal because of this. And this is very typical for fluorine MRI. And it's actually the reason why for us it's so important to always play and to play with the particles we obtain, and the morphology of these particles is very important for us. You can also see that on the relaxation times again, specifically for the T2 relaxation time, you can see that it's almost double compared to the micellar-like system. The physically cross-linked nanogels always have way more suitable T2 relaxation times. So one of the applications of these polymers is called thermogelling polymers. It's actually quite well known in literature. So what you can do with these polymers is that you can co-inject them together with the drug. And the idea is that you should be able to prolong the release of the drug that have been already described. So we wanted to test it with one of the polymers we made. And we use Psi-7 fluorescent dye as like a model drug to actually see what's happening with the drug. We were able to see that when we injected with the polymer, we almost doubled the half-life. So that was already very interesting. Or that's something we actually expected. But what we didn't expect is that when we actually measured the polymer, what's happening with the polymer. So what we expected is that we will see a depot from which the drug will be released over time. But when we checked the polymer with the fluorine MRI, we found out that the depot is gone within 24 hours. So there is no depot, which would be releasing the drug. So actually, the reality is that the reason why the half-lives of the drug extend with these polymers, it's unknown. And we propose that maybe the idea is that the polymer takes the drug inside the cells, and that's maybe the reason. This is just an image which you already saw. It was like our idea. This is what we wanted to see actually with the system. But what we found out is that the cyanine-7 dye goes inside the cells also very quickly. It actually goes faster than when it's with the polymer. So this theory that the polymer could take the drug inside the cells, and that's why the half-life of the drug changes, is not really truth. And we are here a little bit like in that end. We don't really know what's happening. So this is something we will still continue actually investigating. We also made nanoparticles. This is, I will just briefly say it, with the ROS responsive, if somebody would be interested for their specific applications. We made nanoparticles which has ROS responsive monomers. This can be used for systemic drug delivery to cancer tissues or inflammation. We are also now looking into the possibility to use these materials for wound healing. And what happens with this monomer is that when it's oxidized, it basically just decomposes into acrylic acid or methacrylic acid, depends on which material you use. And that completely disrupts the amphiphilicity of the polymer, so it should break apart. Actually, this group is selected because it's very selective to minute amount of hydrogen peroxide or any oxidative reactive oxygen species around 1 millimolar concentrations, actually, which is really ideal because that's more or less the concentration for the pathologies. So if somebody would be interested, we have these particles and we still think about actually how to test them in vivo. So now I will move to my last projects. Those are the chemically crossing materials, which can be then used for tissue engineering. So I will briefly just introduce the regenerative medicine because I believe that probably majority of you know this method. So in regenerative medicine, we can harvest patient-derived stem cells, or this is the ideal scenario. You can add specific biological clues. Then you can seed them on top of your biomaterial scaffold. And the idea is that these cells will grow on your biomaterial scaffold, so it has to be cell interactive. And then when you implant it back to the patient, the material should degrade slowly and it should be replaced with the specific tissue which is growing. So it's very important actually that the degradation rates perfectly matches the growth of the tissue. So that's something which that's the reason why we are so it's so important for us to actually measure the degradation rates. Also, another thing I will be talking a lot about later is mechanical properties of these scaffolds should always match the mechanical properties of the specific tissue. If there is a mismatch, the cells will not grow, so that's also very important for us. So the current approach for vast majority of biomaterials which are tested for tissue engineering is to take large groups of animals and then sacrifice them in a specific time points, dissect the material out, and then you usually also obtain relatively limited amount of data because the materials are usually soft or brittle, and it's very difficult to actually obtain a proper information about what's happening with your scaffold. So, of course, way better strategy is to do the in vivo imaging, but it's not really so much explored yet. So the first group of material I will be talking about are based on gelatin. Gelatin comes from extracellular matrix, so it's inherently cell interactive. We modify this material with a photocross-linkable group. So when they are cross-linked, you can obtain these cross-linked hydrogels. And this can also be used in light-based 3D printing methods. So we can 3D print a specific design, which is important for us. We always print porous structures because without pores, the cells cannot migrate. And the pores are really determining also if the tissue engineering will be successful. So one of the most common materials we are actually using is so-called GELMA, is when you take a gelatin and you modify the amines of the peptide with, you transfer them into methacrylamides. And also another approach, which is a little bit more recent, we are doing is that we also take the carboxylic acids of the peptide to further modify these materials with aminoethyl methacrylate, AIMA, we call it JELMA AIMA, and then you create material which is even more, having even more cross-linking groups. So when you cross-link it, the cross-link density is higher and you should have less swelling and stronger materials. So when you cross-link these materials, you can use light-based initiators, which are sensitive to a specific wavelength. Then you irradiate them and you create basically these polymeric linkers between the gelatin chains, and you obtain your hydrogels. So the first idea, which we thought is basically the most simple idea, just to test this specific system, was to, during the cross-linking of gelatin, to add the fluorinated monomer. So when you cross-link the material, you will obtain this hybrid structures where you have your synthetic polymer combined with the peptide, with the gelatin. And for that, we also can use, here I write it as this degree of substitution, different degree of substitution of gelatin. So you can modify, here I say 30, 70, and 100% of amine groups. And this results in different cross-link degree of the gelatin and different stiffnesses and different swelling. So this is something how we can kind of play with the stiffness of the material, also with the degradation. Actually, it affects the degradation a lot. So here is just the most simple, like a summary of, I think, most important results. First of all, I would want to say that what I did in this project, just the first trial, was to simply add the monomer together with the gelatin. And with this approach, I actually artificially increased the concentration of the hydrogel. So this is something which, if you look then here, these three materials are control groups. So it's gelatin without the monomer, and here you have the fluorinated monomer. So you can see, for example, G prime, which reflects on mechanical properties, almost doubled always when I added the monomer, and that's because I use this specific method. And also here you have the swelling ratios, so you can see that the swelling ratios significantly dropped for when I added the fluorine. So this is something I would say not really ideal. I wanted to add the tracer without affecting the mechanical property, or in general the properties of the materials. But I will show you later how we actually solved this. It was not so problematic at the end. So in order to see, we wanted to do a simple in vivo test where we actually implant these materials and measure the degradation kinetics. So for that, we chose to 3D print these materials into porous scaffolds already. We have many 3D printing methods, but I chose, I would say, the most simpler one. It's called indirect 3D printing. You first print your, it's called sacrificial mold. We are using usually PLA. Then you soak the sacrificial mold in your solution. So you have to also fill all the pores. So I use vacuum to fill all the pores. Then you cross-link it, and then you basically dissolve the PLA with the chloroform, and you obtain your 3D printed materials. This is like a strategy which you can actually, anybody can use it. It's very simple. And you can also make relatively large scaffolds with this, with no optimization of your ink. So that's really nice. So we implanted these materials in vivo. We had these three materials. We actually could see a little bit the scaffold already with the proton MRI because it was porous. So we could little bit see the porous structure. But of course, with the fluorine MRI that added way nicer image, we could see we could determine the volume of the scaffolds. What I was a bit sad about is that because the resolution of fluorine MRI is not so great, I couldn't really see the porous structure in the MRI. So that was a little bit not really so ideal. So we measured the degradation I just want to tell you that normally cross-linked gelatin degrades something between weeks to several months. So you can already see that this is the time period of 1 year. 2 of our material did not degrade whatsoever. So this was really undesirable. And so we can see that really the addition of the tracer impacted these materials. But fortunately, one of our materials was able to degrade. So we kind of know more or less where the threshold is of the amount of cross-linking groups we can introduce into these materials. And we can actually even go, let's say, lower. And with this, we can even introduce some little bit of tunability, because sometimes the degradation of gelatin can actually be too fast when you have unmodified cross-linked gelatin. For some applications, it's really not desirable. So there is a need for materials which degrade slower. So that's something we want to also later integrate. What we also found out, if you look at the histology, this is basically all these purple parts are the hydrogels. And what we found out that in the places where the material either degraded or where we had the pores, they were all filled with adipose cells. And that was really nice to see, because the possible application, usually the targeted application for these materials is actually to do adipose tissue engineering for breast reconstruction. So we were quite happy that we see that the adipose cells from outside migrated in our scaffold, because they liked the environment and they could grow on it. So as I said, this method where I added the crosslinker significantly increased the mechanical properties and slowed down the biodegradation. So in the next study, if you would want to see more details, here is the reference. What I did is that, because we know that the gelatin, we can really play with concentration and we affect the properties of the resulting hydrogels significantly. So what I did is that with the addition of the monomer, I was decreasing the concentration of gelatin. And then I actually found out I can make materials which are biodegradable, and they have very similar mechanical properties. So this was very nice. The only thing we were scared is that if we limit the amount of gelatin in the structure, it will stop being cell interactive. So then we confirm that indeed our materials are actually still cell interactive, even though they sometimes have even 1% of gelatin and less. So that was really nice, actually. So and now I will also talk about the contrast agents for radiology. So as I said in the beginning, we had this moiety, which is very known to be a building block for contrast agents which are used in the clinics. And that we transform into the metacrylamide to get this monomer. And we did basically exactly the same strategy as we did before with the fluorine. So we added the monomer directly during the cross-linking. Since here we were adding, we used the same method, but the reason was that here we are adding approximately 20 to 40 times less of the monomer than for the fluorine, because the radiology is so much more sensitive, let's say. So we didn't really worry about affecting the mechanical or affecting the properties of the gelatin so much. What you can see here, for example, is cross-linking kinetics. So the black is the control without the monomer, again here, and the colorful ones are when we add the monomer. So what happens here is that the cross-linking kinetics are faster, so that's good for 3D printing. But let's say the final mechanical properties, you can also see here, are more or less within the same range. So that was nice for us. What we found out is that we measured degradation in vitro. We found out that for the materials based on gelatin metacrylamide, we were able to create materials which degrade similarly as the control, which is again black. But we found out that when we add the monomer to the gelma-AIMA, which is the material with even more cross-linking groups than gelma, no matter, it doesn't really matter how much of co-crosslinker you add to that hydrogel structure, it will immediately became non-degradable. So that's something, yeah, we really, if you want to have these materials degradable, you cannot basically add a co-cross linker. So that's something which we cannot affect anymore. Here you can see how it actually looks like. I add these are two controls. I put crushed hydrogel into a Eppendorf tube and I put PBS on top. And you can see nicely that with the more of contrast agent I add into it, the higher is the contrast. And here we also compared it to tissues. So you see that the controls are somewhere in this, let's say, gray zone, while when you put the contrast into your materials, you obtain contrast comparable to bones. So this is already giving us an idea that we will really be able to really nicely see the materials in vivo. So we took several of these materials. Again, we implanted them in vivo. On one side, we implanted again a porous scaffold. And on the other side, we decided that we could also test another approach where we take our hydrogel, we dry it, we blend it. Then you can also sieve it to have a specific sizes of nanoparticles. And then you can reswell these nanoparticles and inject them. So the interesting part about this approach is that the nanoparticles are never very smooth. So they actually create specific pores, like pores between each other, like gaps between each other. So the cells actually really like this environment. They can really nicely migrate through these particles, and we also call it injectables, these materials. So we actually, during this implantation, we actually had some issues with these materials. You really have to test it several times, I think, to get it right. So what we saw is that we have air bubbles. That was one thing, but that was not such a problem because the air bubbles usually disappeared within few days. But what we also saw that this the material could spread all over the place. It didn't really stay as a little compact scaffold. And I think with one group, we were even injecting water for all of the samples, and then we even had to remove this group from the results. So this is a little bit annoying, but I also find it quite interesting that the fact that we have the possibility to measure the CT directly after the injection, it helps us to know what's happening and if we are supposed to actually maybe reapply the materials. I think next time if we would be doing this and see these defects, we would probably try to reapply the material again. So here is a little summary what I want to see here, show here. I will later show process data, but I only wanted to show you that, I don't know if you can see it, that for example, for the porous scaffold, we could really see a collapse of the pores. They start disappearing. So this is something we will have to, for example, investigate with cells if this is actually suitable. And then another thing which we found out is that we can measure these materials also with ultrasound because the material is so porous and different compared to the other tissue around. We can really nicely see it. And this is very nice because like this, you could theoretically limit the exposure of the patient to the radiation. So this could be really nice. Here we have the results from this in vivo study. So I will be first talking maybe about the volume. What I find really nice about this is that we can really nicely see from the results the mechanism of the degradation. So in the beginning, you can see that the volume increases here up to the day 20. And that we think that it's because the material swells because you have a bulk degradation. So that means that the material, the cross-linking points of the material inside the material are breaking. That's why the material is swelling. So that's called bulk degradation. And that at one point, the materials volume started rapidly falling down. So here we didn't know if it's bulk degradation or surface degradation. So what we look at is average radio opacity during the measurement, and you can see it's fluctuating a lot. But what you can see that in the beginning and at the end, the radio opacity of the material is more or less the same. That means it doesn't really changes over time. And so what we think is that the material is only basically degrading from the surface. So it's just getting smaller and smaller and smaller and compact, but the radio density doesn't change. So it's not basically diluting itself. So that's really nice to see. And this is completely last project I will be talking about. I still have 3 minutes. So these materials are a little bit different. They are based on polycaprolacton. Polycaprolacton is hydrophobic polyester. It's completely not water soluble. So when you cross-link it, you will obtain materials which are very dense and hard. So actually, they can be used for tissue engineering of bones. So this could be really beneficial to also integrate the contrast agent into these materials. So here is just, again, similar cross-linking strategy. The chemistry is a little bit different. Here we are, instead of acrylates, we are integrating a thiolene chemistry. And again, that actually can be combined with it can be combined with the acrylamide monomer. So first of all, I would like to show you, this is Young's modulus of these materials. And this, again, here is 0 amount of the monomer. So that's the control. And you can see that the mechanical properties of these two controls are really remarkable. It's between 50 and tens to hundreds of megapascals. So that is really matching actually mechanical properties of trabecular bone. So that could be used for tissue engineering of some small, let's say, bone defects. Also, another thing about these materials is that they have really amazing elongation break. That means that how long you can stretch it until it breaks. So for the materials, it's usually actually around 500 to 700% stretchability. And so you can actually imagine that I really didn't want it to affect these mechanical properties with the addition of the monomer. So this is what I was measuring first. So when I add the monomer in different ratios, is it affecting the mechanical properties? And I think I only found this specific number which was significantly different. And that was actually the highest ratio between the monomer and the alkene groups in dark black here. So I kind of established, let's say, thresholds until I can go with the contrast before I start changing the mechanical properties. And also what I want to show you here, I'm actually adding weight percent. I write it in weight percent of ATIPA because this material doesn't swell, so you don't have to add a lot. So I usually add it around here 40 milligrams per 1 gram, more or less, 80 milligrams per 1 gram. So that's relatively little. And you will see that it really, the radio opacity of this resulting material is really high. So again, I compared these materials with different tissues, and you can see that with the 16 weight percent of the monomer, you could go to 3,000 Hounsfield units. So I think that's a bit exaggerated. You actually don't have to go so high. So these materials could be really used already. And you can also see it's really nice because the material doesn't swell. You can really put a linear fitting between the more you increase the iodine, the more increase the radio opacity, but the relation between this is perfectly linear, so it's really nice to see. So what we did also is to show that we can actually visualize nicely the scaffold. So we again make a negative scaffolds through indirect printing. And this is the scaffold with the contrast agent, and basically the scaffold without the contrast agent, we were not able to see in the body whatsoever. We couldn't localize it. Then we looked what happened after half a year exactly. with the scaffold, and we saw that the volume of the scaffold decreased to 40%. So we also show that these materials are degradable. And based on that, we decided to print materials. I decided to actually employ a little bit more advanced 3D printing. This is called volumetric 3D printing. Here it's relatively new technique. You are projecting 2D images through a rotating vial. So you're able to print the whole structure of the implant at once. So you can actually make these kind of structures within seconds. So that's really nice. So then we implanted these materials again in vivo. We selected two materials, one with 4 weight% of ATIPA, one with 8 weight% of ATIPA. And we were measuring again the degradation kinetics, but this study is still ongoing. We are now at month 7, I think. And we still decided to do another 2 time points. So we will see what's going to happen. But what we actually see here, what I wanted to show you, maybe a bit interesting, is that we see that the average density is really falling a lot. So what we believe is that these materials actually follow a bulk degradation all the way. So basically the material is maintaining its structure. After all those months, it's still porous, it's still maintaining structure, but it's getting less and less and like more diluted inside. So that's something which we found quite interesting. The problem is that for us is that we are a bit scared that at one point we will not be able to see the material anymore. So that's the only thing which we are a bit scared about. But let's see. So with this, I would like to summarize. I just want to say that which I didn't really mention so much is that the materials which were supposed to be cell interactive were fortunately cell interactive, and that all the materials we developed were biocompatible and also well tolerated in vivo. So that's really nice. And another thing we always look at is that if they accumulate somewhere specifically and they do not. So that's really nice. And with this, I would like to thank mostly my colleagues from TSAPE who are measuring all the in vivo studies, and also Euro-BioImaging for all the support of basically all of the in vivo studies. Yeah, okay. Thank you very much for your attention.